Hi everyone, and thanks so much to OHBM for this incredible opportunity to present my research. Today, I plan to propose a roadmap towards precision cortical imaging, where by this I mean the development of tools for tailored diagnosis, prognosis, and mechanistic modeling of a range of neurological processes within individual human brains. I plan to discuss some key methodological barriers which I believe are holding the neuroimaging community back in this respect before introducing some tools that myself and my team have been developing in order to address some of these challenges. So, what does precision neuroimaging look like to me? Well, what I think we'd all like to be able to do now is to take some individual and put them in the scanner, where this here is my son Ash volunteering for the Developing Human Connectome project. From them, we collect multimodal imaging data, clinical background information, demographics, and sometimes genetics. And then we seek to push these through learning models and simulations in order to get back highly accurate predictions of what the likelihood is that that individual either has or will go on to develop some cognitive or clinical phenotype. However, ultimately, in an ideal world, what I think the long-term objective should be is to look towards building mechanistic models of these processes tailored to individual brains, from which we can then model interventions and treatment strategies. So clearly, there are many obstacles and challenges in the way of achieving such a vision, but the one I'm most interested in currently is the question of how can we most accurately compare brain imaging data across populations in order to truly know what is normal for any given individual and build simulations tailored to their brains. And this is made inordinately more complicated due to the breadth of variation of human cortical organization, where the cortex, as we all know, is this thin layer of tissue at the outer surface of the brain, which forms complex convolutions in order to make room for a dense network of interconnected cells, which mediate complex patterns of dynamic brain activity, all of which underpin many aspects of cognition that we're interested in. Classically, human brains have been analysed at the population level by first diffeomorphically or smoothly and invertibly deforming input brain volumes until their shape more closely resembles that of some target or reference brain, at which point we assume all images in our population completely overlap, which allows us to fit statistical models either at each voxel or, if we're feeling a bit more conservative, within individual regions in order to allow us to compare differences between groups of individuals. Why do we do it this way? Well, comparing human brains is what's known as an ill-posed problem, which means that there are many different ways that we can map a voxel or cluster of voxels in one person's brain into the reference space of another's. And so that means that we need to impose some form of regularization so that we learn a realistic or biologically plausible solution. And on the face of it, diffeomorphic mappings make sense because they enforce that voxels must deform smoothly along with their neighbours. And this ensures that we don't just end up with some jumbled up jigsaw of one person's brain in the space of another's, which is clearly not what we want. So in more general terms, if we consider these circles on the right-hand side to be brain regions, then the types of deformation that we're allowed under a diffeomorphic constraint are that we can non-rigidly grow or shrink our brain regions, meaning that each brain region can deform in a different way to its neighbours. And this includes moving some regions closer together and others further apart. But what we absolutely cannot do is swap regions around or split them in two. And the problem is that when you really start to look at the types of variation that can be observed across human brains, then you start to realise that this just simply isn't enough. For example, taking these three core-scale representations of cortical folding here, where yellow represents gyri and blue sulci, then you start to notice that these vary in ways for which it's not immediately clear what the most appropriate diffeomorphic mapping should be. Particularly within the frontal lobe, you'll see examples where one individual will have multiple folds where another individual will only have one. Similarly, if we start to then look at fMRI mappings of cortical areas, we start to see types of variability that would require mappings which swap the locations of regions around or split them in two. Transformations which, as we've just said, are completely prohibited under a diffeomorphism. So this is just one example taken from the HCP cortical areal parcellation, where this complex highlighted in the red box here indicates the orange region 55B, which is typically bordered either side by the frontal and posterior eye fields. But in a sizable subset of individuals, we see that 55B is split and the posterior and frontal eye fields merge. However, this is far from an isolated problem specific to the HCP parcellation. Examples of topographic variability have been repeatedly reported within the cytoarchitectonic literature. Other examples have been reported from analysis of fMRI of the fusiform face area and early visual cortex, and the need for transformations that locally break diffeomorphisms have been repeatedly called for by the fMRI decoding community, with groups such as the Haxby Lab advocating for hyperalignment methods which abandon this constraint. Moreover, topographic variation is not the only problem to contend with. So we also need to be mindful of evidence that suggests a dissociation between patterns of cortical shape and function across many areas of the brain. Where perhaps the best example of this was described in a FreeSurfer study in 2008, which performed shape-based alignment of 10 brains scanned post-mortem, where for each of these brains they had also obtained ground truth segmentations of cortical areas using histological dissection. And the results showed that while overlap of cortical areas was good for many primary areas of the brain, higher order regions such as Broca's, which is highlighted here in the pink box, these show much poorer overlap, suggesting that the location of folds is really not a good marker of the placement of these regions. So what are the downstream consequences of ignoring these problems? Well, residual errors in alignment are going to add considerable noise and uncertainty to downstream analysis, muddying interpretation and obscuring subtle signs of disease. Where evidence of this can be seen by looking at this example of a normative model of cortical folding development presented by my PhD student Yuran Guo at OHBM this year, where in this instance, cortical folding maps from all individuals from the Developing Human Connectome project were first mapped to a population average reference space, and then a separate Gaussian process regression model was fit at each vertex, thereby summarizing cross-sectional changes in cortical folding across gestation at that location. So in this way, we were able to generate a high-resolution spatio-temporally continuous surface template. But what we need to notice is that as cortical folding is developing, the heterogeneity in the normative population is increasing uncertainty in all of these models. And moreover, this bottom plot is highlighting a population of outliers, which indicates a subset of individuals that just happen to have a rare gyrus when the majority of individuals have a sulcus. And importantly, I would argue that this is not just a problem at high resolution, but is going to generalize to models fit at cortical areas, since if we don't know that we're doing a good job of aligning regions using shape-based alignment, then we can't know for sure whether outliers from normative models are genuinely reflecting phenotypically relevant variation or are just picking up on image registration error. And so ultimately, all this is reducing our statistical power at the cortex to detect subtle features predictive of cognition or disease. Further, this isn't just a sensitivity issue, but also one of interpretability, where this was highlighted nicely by studies by Janine Beisterbosch in 2018 and 2019 that showed that through a combination of canonical correlation analysis and simulation, that up to 62% of the variance captured in functional connectivity matrices might actually just be encoding residual misalignment, essentially meaning that a lot of behaviorally relevant variation is remaining encoded in resting state spatial maps. But some of this is inadvertently getting pushed into functional connectivity matrices due to either limitations of dual regression or perhaps through the use of group average parcellations, which is creating an appearance of differences in functional coupling when actually the problem is that we're not estimating functional connectivity between the regions that we think we are. So bearing all this in mind, what can be done to try and improve the precision of our analysis? Well, one thing that I would recommend to reduce, if not eliminate, misalignment is to move analyses from the volume to the surface. since putting aside for one moment concerns over dissociation between function and structure, which I will address in the next section, there is ample evidence that moving from volumetric shape-based alignment to surface shape-based alignment is already improving the correspondence of functional areas, where this was again shown nicely through the free surface cytoarchitectural study that I talked about a few slides time ago. And likely this is because surface diffeomorphisms and smoothing operations are regularized based on geodesic distances along the cortical sheet rather than Euclidean distances in 3T, and that means that they are better respecting or preserving patterns of cortical organization. So in our work, image registration is driven using multimodal surface matching, or MSM, which was a tool that I developed when working for the Human Connecting Project. So like many cortical surface registration algorithms, MSM works by first projecting cortical anatomies to the sphere, where it does this to help simplify the matching problem from one of matching complex shapes in 3D to one of instead moving points around on a source or moving mesh until some generic pattern of cortical organization on that mesh more closely overlaps with equivalent patterns of organization on some target or reference mesh. And then MSM is set up with a flexible discrete optimization framework, which is essentially minimizing a data term whose job is to encourage displacements that are increasing the overlap of those structures in the source and target mesh, but balancing this against a biomechanically constrained hyperelastic strain penalty, whose job is to encourage a smooth and biologically plausible diffeomorphism of the sphere. But importantly, this regularization can actually be imposed either on the local affine transformations of this learnt spherical warp or the implied transformations of the anatomical grid, where I'll come back to the importance of this in a few slides' time. So to first explore the benefits of MSM simply with regards to shape-driven alignment, let's consider some examples from our Developing Human Connecting Project cohort, for whom we have developed bespoke surface extraction pipelines to extract high-quality cortical surfaces from both our neonatal and, more recently, our fetal cohort. Where here I'm highlighting very recent work presented at OHBM by colleagues from King's College London, which describe spatio-temporal surface and volumetric fetal brain templates, which are summarizing population average properties of brain development across late gestation for these babies. These templates actually reflect the culmination of a huge body of work that went into getting our fetal data sets reconstructed, motion corrected, and surface extracted in the best possible quality, where in fact these surface templates were generated from cortical segmentations that had all been painstakingly manually corrected in response to some initial challenges that we faced in translating our neonatal segmentation tools to our fetal cohort. However, fortunately, this has now been addressed through using these corrected segmentations to develop and train a novel automated deep learning segmentation tool, which importantly generalizes across acquisition protocols and gradient strengths. Now, looking at some downstream applications, we can start to see examples of where surface analysis is really offering unique advantages over volumetric analysis for studying certain properties of the cortex, in this case cortical asymmetries. So asymmetries are features of cortical organization that are implicated in a wide range of cognitive processes as well as many neuropsychiatric disorders. And they are best studied on the surface because this allows for data to be mapped to a symmetric population average template, which has been engineered to have interhemispheric vertex correspondence, essentially through taking templates for left and right hemispheres, co-registering these and then averaging them. And then this therefore allows asymmetries to be simply calculated for each individual by mapping their features to the population average template space and then simply subtracting the left and right hemispheres. So this approach then allowed my PhD student Logan Williams to investigate genetic versus environmental origins of structural and functional asymmetries using a cohort of healthy term-born babies scanned shortly after birth, where these were then compared against a smaller cohort of moderately preterm babies scanned at term equivalent age and a group of young adults from the Human Connecting Project. In this way, Logan was able to show that many of the asymmetries known to exist in older cohorts of adults and children are indeed present at birth, suggesting that they're genetically encoded. And interestingly, there were minimally statistically significant effects associated with moderate preterm birth, suggesting that these factors may, to an extent, be robust to early adverse environments. And here we see another potentially useful feature of MSM, which is the integration of hyperelastic strain regularization over the implied anatomical warps, since this allows us to impose a biomechanical model on our longitudinal registrations, where this was applied by colleagues at WashU to study cortical growth of a small cohort of very preterm babies that were scanned at regular intervals between 28 weeks and 38 weeks gestation, where in this case, anatomical MSM registrations were used to register between time points within each individual in order to output estimates of areal expansion at each vertex, which could then subsequently be compared at the population level by mapping all data to a population average template, allowing us to look for common developmental trends. And these results were interesting, showing that growth across the cortex is not even, but rather is initially fastest in the somatomotor, visual and insular regions, but later rates of growth in these areas slow down to be overtaken by growth in the temporal lobe. And more recently, we were able to reproduce some of these findings in our DHCP fetal cohort by similarly fitting an exponential model of growth to longitudinal anatomical MSM mappings, which this time had been estimated for individuals that have both a fetal and neonatal scan. However, in this instance, we had no control over when the babies were scanned, so we model age at scan as a covariate. Results here showed that individuals that were scanned earliest in gestation and therefore had the longest scan intervals were showing faster rates of growth in the somatomotor and insular regions, replicating the results from the previous study. We then sought to compare in utero growth to ex utero development of preterm neonates over the same period, finding differentiating rates of relative growth in the posterior lateral parietal cortex for fetuses, but in the temporal and frontal poles for preterms, where this observation of elongated brains in preterms has been previously reported in the literature. So this clearly shows benefits of surface analysis for evaluating morphological processes, but the next thing we can start to consider is the question of how can we address the residual dissociation of cortical function and structure in order to more accurately compare cortical areas across individuals, where for this we can leverage the full functionality of MSM and perform multimodal or areal-driven alignment. And further, as we will see, these benefits can be even more clearly realized when we address residual misalignment through the use of transformation invariant machine learning frameworks. Where for the HCP, our preferred configuration of functionally driven MSM is more commonly known as MSM-all, where this drives alignment with a multimodal combination of T1 over T2 weighted myelin maps and core scale surface RSNs, where this specific configuration was chosen as it was found to optimally improve the overlap of cortical areas as validated using independently collected task fMRI experiments, as we can see here on the right for a language and working memory task. So this shows on the top here group task statistics following cortical folding driven alignment, and on the bottom group task statistics following areal feature-based or MSM-all alignment, and we can immediately see evidence of improved overlap in functional areas for MSM-all as improvements to the group task statistics, which are seen as increases in the extent or brightness of yellow clusters, which reflect increases in the group task statistics. Setting up MSM in this way was fundamental to the development of the HCP multimodal cortical parcellation, since it allowed for generation of sharp group averages of multimodal features, including task contrasts, resting state ICA maps, cortical folding, cortical thickness, myelination, and visuotopic maps, where these average maps were then hand annotated by David Van Essen and Matt Glasser, drawing boundaries at locations where they observed sharp changes in contrast that were consistent across multiple modalities, allowing them to generate a group average map of cortical organization, which closely resembles known patterns of cytoarchitecture. Where then residual misalignments were subsequently addressed by training vertex-wise classifiers, one for each region, to learn multimodal fingerprints that essentially describe the expected tissue properties at each vertex. For example, each vertex in the region 55B, highlighted here in the bottom panel, would be expected to be represented by a feature vector that describes high values for language RSNs and language tasks, but low values for myelin and for the upper limb RSM, where here we can see that it's falling in this small gap. So one challenge with training such learning models is that we only had one set of labels for the group average. So this was addressed by filtering training examples from the full dataset used to build the group average map to retain only examples whose imaging features strongly correlate with that of the group at that region. And this could then subsequently be used to train those classifiers that could detect those regions in atypical subjects like those shown on the right. Importantly, we now know that this pipeline generalizes from HCP quality resting state data to datasets with much more standard fMRI protocols, where here I'm showing MSM all validated for UK Biobank, which collects only 6 minutes resting state fMRI relative to the hour collected by the HCP. Nonetheless, we see strong improvements in sharpness of language RSNs and their gradient maps as we move from MSM Sulk to MSM all, particularly picked up here in the gradient maps. And similarly, validating on independently collected task data, we're also seeing improvements in the group task statistics with more regional clusters appearing, as we can see here. So this then puts us in a position to start thinking about how to propagate the HCP parcellations to individuals from the UK Biobank. However, initial efforts in this direction that looked at direct application of the HCP vertex-wise classifier were not at all successful, where this was largely, I think, due to much higher levels of noise in the fMRI spatial maps. And so for that reason, we started to look towards convolutional neural networks, since convolutional neural networks offer this theoretical advantage over classical machine learning in that they can learn transformation invariant representations of images, which importantly means that they do not require images to be aligned at all in order to compare them. Rather, CNNs work through learning filtering operations, where filters are small grids parameterized by weights, which importantly are learnt during training through translating filters across the surface and then comparing them against image features at each location so that they can learn to become sensitive to different textures in the image. And then complexity of these filters is built up over many convolutional layers, such that filters from early layers essentially are acting as edge detectors, whereas later filters are activating for individual objects. And in this way, image understanding is then broken down into a problem of essentially just asking what nonlinear combination of filtering operations best describes each image. At this point, however, I do want to emphasize that translation of convolutional neural networks to surface domains is really non-trivial, since surface manifolds have no global coordinate system on which to constrain a transformation of a filter across a surface in a consistent way. And so actually, the only mathematically correct way to implement a convolution for a manifold is in the spectral domain using generalized Fourier transforms. However, this is often prohibitively computationally costly. As a result, there is not one way of implementing convolutions on a surface or a graph. Rather, a number of different approximate solutions have risen up, where each of these are essentially trading off properties of transformation invariance against network expressivity, meaning that you can't typically achieve both completely, unlike Euclidean CNNs, where I really can't understate the importance of these two properties. They underpin much of what has made Euclidean CNN so powerful. So for these reasons, prior to developing any tools for cortical surface deep learning, we first benchmarked the performance of a number of different geometric deep learning frameworks on the tasks of cortical segmentation and phenotype regression using data from the Developing Human Connecting project. And I really recommend anybody wanting to get up and running with these tools to check out Abdullah's paper and potentially make use of the accompanying code. However, at a high level, the key takeaway that we found was that we really couldn't compromise too much on filter expressivity if we wanted to learn complex cortical imaging tasks, but nonetheless, you have to bear in mind that cortical imaging datasets are small in deep learning terms, and so invariance to rotational transformations is going to be necessary in the long run if you want to learn parameter efficient and interpretable solutions which generalize and don't overfit to your data. So, informed by all of this, Logan developed a surface convolutional neural network architecture to robustly propagate the HCP parcellation to individuals from UK Biobank in this work that's presented at OHBM this year. And he achieved this by training a separate surface convolutional UNet network for each cortical region, using the HCP individual parcellations and their corresponding feature maps as training data, but refining the feature set to remove task fMRI as this was not available for UK Biobank. And in this way, he generated greatly improved parcellations relative to a vertex-wise fully connected neural network, or FCNN classifier, which itself represented a highly optimized network relative to the one originally used by the HCP. Where even the most problematic region in the brain was being detected in 80.9% of all of the 3,000 UK Biobank subjects tested, relative to only 29.9% for the fully connected neural network classifier. And this included specific improvements to areas such as the insula, where here we can see from the white arrows examples of regions that were missing from the group-wise parcellation generated from a vertex-wise classifier, which are now being successfully picked up in the group average parcellation for the convolutional approach, such that the group average UK Biobank parcellation is now really closely resembling that of the group average HCP parcellation, and that was true across the entire brain. However, importantly, this is not because the network is assigning a group average parcellation to all individuals, because we are still seeing significant residual intersubject variability, including evidence that the model is still able to capture atypical topographies of 55B, as well as generating parcellations which are largely consistent over time, despite the effects of healthy aging which occur over the relatively large time intervals that occur between the repeat scans collected by the biobank. Similar benefits in using surface CNNs have also been seen by collaborators at UCL in their development of graph convolutional neural networks for detection and segmentation of focal cortical dysplasias, where FCDs here are known to be the leading cause of drug-resistant childhood epilepsy, but reflect extremely subtle lesions that can be very difficult to visually detect on structural MRI, even for very highly trained clinicians. So ultimately what this means is that ground truth annotations that are used to train automated segmentation tools suffer from significant inter-rater variability and uncertainty, but nonetheless, the proposed graph convolutional UNet network does a very good job of predicting lesions within 20 millimeters of these masks, again significantly improving over a vertex-wise fully connected neural network classifier, in this case otherwise known as an MRP, particularly in terms of outputting contiguous predictions with far less false positive detection spread out across the cortex, where this is something that's really important in terms of increasing clinical confidence in the utilization of such a tool. Moreover, deep learning is not just for phenotype prediction and segmentation, it can also be used for other image processing tasks such as image registration too, where here I'm showing a learning-based implementation of MSM developed by my postdoc Mohammed Suleiman, known as DDR, which learns mappings in a completely unsupervised way using image similarity measures to drive optimization before outputting a spherical warp which is smoothed with a deep conditional random field, to generate mappings that are both smoother and better aligned than the majority of existing classical surface registration algorithms and all previous learning-based approaches, where here we see that DDR is outputting much smoother warps than a competing learning framework known as S3Reg, equally as smooth as the hyperelastic strain regularized version of MSM. Whilst generalizing to new data sets sufficiently robustly that the alignment of test subjects is better than MSM and almost as good as spherical demons for cortical folding. But unlike spherical demons, newer versions of DDR, known as geomorph, also generalize to multimodal registration, generating sharp group averages of cortical myelination and function that align small and highly variable structures in the images as well as MSM all. However, ultimately, really the reason why I'm interested in the potential of fast learning-based image registration frameworks is that, in my opinion, they present the perfect testbed for designing entirely new image registration paradigms that, for example, might support integration of generative models of cortical variation, on which we can also start to explore rapid prototyping of new regularization constraints that might break free from the rigidity of diffeomorphisms. Where here I'm just presenting one baby step in this direction taken by Yurong in this work presented at OHBM, which used DDR, because it's so fast, to co-register all pairs of HCP subjects in a way that it was then possible to get back really accurate estimates of the overlap and therefore the similarity of their cortical folding patterns, which in this way was then used to cluster out individuals that share common patterns of cortical organization in order to co-register these hierarchically by first aligning individuals with the most similar brains before incrementally aligning more and more different brains until everything is brought into a common reference frame. In this way, we are then able to pull out common variants of cortical folding, shown here for the frontal lobe, which replicate a number of patterns previously reported from observational studies, but I think importantly now this is providing us a way of investigating these traits at scale using big data sets such as the HCP or UK Biobank to explore genetic or phenotypic associations. So finally, let's discuss how deep learning can allow us to go beyond discriminative predictions towards tailored explanations for decisions within individual brains, or even build mechanistic models of cortical processes. So one major recent development in computer vision has been some move away from convolutional neural networks to what are known as vision transformers. Transformers are classically thought of as tools for natural language processing. They work using a powerful concept known as self-attention to model how words and phrases work together to convey meaning, where importantly these associations learnt through attention can be complex, long range, and in this way integrate information across an entire body of text. So the motivation behind vision transformers, therefore, was a realization that comprehensive understanding of images also requires integration of information across a scene. Where, for the particular example of brain images, I think we can probably all think of numerous clinical diagnoses that require a holistic picture of the entire brain, which is something that just isn't really afforded through convolutional neural networks, which only learn local representations. So vision transformers therefore work by taking an image and patching it up into sections and then flattening those patches and treating those patches like any other sequence learning problem. Where interestingly, this also helps us get around the problems associated with translating convolutional neural networks to the surface by avoiding it entirely. So in this work, my PhD student Simon Dajon was able to translate the concept of vision transformers to cortices by patching cortical imaging data on a sphere using a regular triangular tessellation of a low-resolution icosahedron. And then in this way, he was able to develop a multi-scale surface vision transformer that strongly outperformed the best performing surface convolutions. Where here I'm showing results specifically for the task of prediction of gestational age at birth, where essentially the objective is to predict how preterm a baby is from their term age scan. And if we look at this final column here, we can see that the mean absolute error of predictions generated from the MSSIT transformer is much lower than the convolutional neural networks. And this essentially indicates that the vision transformer is learning much more expressive representations whilst also being sufficiently transformation invariant that it's performing comparably irrespective of whether we've pre-aligned our data with MSM or we're just using unregistered native surfaces. But more importantly, transformers are what are known as inherently interpretable learning frameworks, since we can look at what features they attend to in order to make their decision. So here I'm showing attention applied to classify how old a baby was when they were scanned. So rows B and C here are showing attention maps for one baby scanned very preterm and another term baby scanned shortly after birth. More importantly, transformers are what are known as inherently interpretable learning frameworks, since we can look at what features they attend to in order to make their decision. So here I'm showing attention applied to classify how old a baby was when they were scanned, where rows B and C here are showing attention maps for one baby scanned very preterm and another term baby scanned shortly after birth. And we can see that not only are these attention maps very specific to each individual, but also that they are picking up on areas that, as we can see, are developing very rapidly over this period of late gestation. However, we can get much higher resolution and human interpretable explanations if we move beyond classification or regression and towards generative modeling. For example, here I show ICAM, which was developed by my former postdoc Shahbaz, which uses an approach known as image-to-image translation to learn what it would take to change the appearance of an image from healthy to diseased, or in this case, progress from mild cognitive impairment to Alzheimer's disease. And in this way, we can then subtract the difference between the baseline MCI image and the simulated AD image to return a feature attribution map, which is essentially predicting where we expect to see new sites of pathology of the brains to progress. Moreover, ICAM can learn to detect all features of disease within individual brains, even those from heterogeneous areas such as the cortex, because the model learns to disentangle class-relevant features of disease from the vast background of cortical shape variation that would otherwise drown it out. And it achieves this by modeling each source of variation with a completely separate latent space. So each image's class can then straightforwardly be altered by taking their brain shape encoding from the larger content latent space and swapping their disease-specific attribute latent vector to one of the opposing class, and then reconstructing the modified image with a generator network. So here we see ICAM validated against ground truth patterns of disease progression that were obtained by taking 2 scans from an individual known to progress from MCI to AD over that interval. These were then rigidly aligned and subtracted to generate the map shown here in red. And in this way, we can see that the disease maps that are being simulated by ICAM much more closely resemble that ground truth than what can be achieved through post hoc explanations for the decisions of classifier networks or competing generative models such as VA-GAN, where the key difference here is that they're not disentangling brain shape from pathology, and therefore they're not as well able to pick up on the more heterogeneous features of disease within the cortex. But we can also go further beyond explanation towards interpretable forward models of disease, where in this example, Mariana da Silva has been working on a learning-based biomechanical model of brain atrophy in Alzheimer's disease, where this works by first using a fully connected neural network to predict regional patterns of brain atrophy from just demographic metadata. So all the network is given is an individual's age, sex, disease class, and some time interval over which we want to simulate disease progression. The output of this network is then converted to a volume which prescribes atrophy for each voxel in an image, where these prescribed patterns of atrophy are constant within each region. And this model is used to constrain a biomechanical model whose objective is then to learn how to optimally align some ground truth baseline scan to a later time point while staying as true as possible to the prescribed atrophy model by penalizing hyperelastic strain deviations away from this. And then in this way, Mariana was able to generate simulated patterns of brain atrophy which closely resemble ground truth, as well as intervene at test time on disease status or years of progression in such a way that it's clearly possible to distinguish between disease states as seen through measurements of ventricular and hippocampal atrophy. And finally, in this last example, I'm showing a conditional generative model developed by Abdullah Fawaz, which simulates how a preterm baby's brain should develop if they were healthy. So the top line of this figure shows a simulation of how one individual's cortical folding pattern would be predicted to change over gestation if they were healthy. And the bottom 2 images at the bottom show ground truth scans obtained for the same preterm individual scanned shortly after birth and then at term equivalent age, where importantly the simulated model is preserving this individual's specific patterns of cortical shape, while simulating increases in cortical curvature and patterns of cell maturation from T1 over T2 myelin maps sufficiently realistically to age the brains well enough to completely fool an independently trained regression network. But more importantly, a metric summarizing the deviation between the simulated healthy image and ground truth follow-up scan was found to correlate strongly with the cognitive outcome of these babies at 18 months. So in conclusion, hopefully I've convinced you that a combination of functionally driven surface alignment and machine learning offers significant potential for improving the precision of cortical analysis, with recent AI advancements in deep structural causal modeling and transformer-based models of cognition likely to result in more and more ambitious learning models in time. Of course, this does not mean that I am advocating for an end to classical approaches based on image registration, whose greater robustness and more straightforward interpretation over learning-based frameworks cannot be denied. In my opinion, these tools should be developed in parallel, with classical tools continuing to provide insights that can be used to constrain and validate learning-based frameworks. So for anyone wanting to access any of the resources mentioned in this talk, I recommend checking out our website, which will contain any links to code repositories that haven't yet made our GitHub page. I will also upload a copy of these slides and a link to this talk there. I want to particularly highlight the availability of a new version of MSM, which was developed by Renato Bosenci, where this has reduced runtimes of MSM down from 90 minutes to 2. We are looking for anybody willing to test that out. And as always with all of our software, we are really happy to support questions through GitHub or the DHCP Neurostars mailing list. So thank you for listening to me. I have no disclosures to make. I'd like to finish by thanking my team and all funders of the work presented here today. We look forward to hearing your questions.